Good morning, everyone. So I'm going to start us off today with, I'm starting a little bit early before 9:15, with a video that is not directly relevant for what we're studying, but a lot of you might find it very interesting. So last time we looked a lot at responses of phase-selective areas of the brain. And we're about to see a video of a man in a hospital bed who's in the middle of some surgery for epilepsy. And he's got some electrodes on the surface of his brain. Specifically, he's got electrodes on the fusiform face area, which is one of the areas that responds most strongly to faces. We're going to see what happens when we stimulate those electrodes and activate his fusiform face area.
Look at my face. 1, 2, 3.
You just turned into somebody else.
Tell me
Your
about
face
it.
metamorphosed. Your nose got saggy, went to the left. You almost looked like somebody I'd seen before, but somebody different. That was a trip.
California.
Could you see my eyes in place?
I could see your eyes, but you could have been somebody else who had, well, he has similar eyes to Dr. Parvizi, but you were someone else. Your whole face just sort of metamorphosed.
Metamorphosis. Did it get warped?
Yes. You looked like someone else.
Did I keep my gender?
Yeah. Oh yeah.
How did you know I'm not a female?
Because you're still wearing a suit and tie.
Oh, you could see the suit and tie?
Yeah. Only your face changed. Everything else was the same. Yeah.
Did the skin color remain the same?
Yes.
Onto the main stuff that we're talking about. So today we're going to look at higher visual processing. So we're going to basically just build up on what we were looking at last time, extending up to higher levels of visual processing. So why do we want to study this? These are generally very similar reasons to last time. Okay, so we study higher vision because image processing is a common application of artificial deep networks, particularly in the case of object recognition, so we're going to focus a bit on that. Also, and this is something we cover a lot today, there are various important properties of early visual processing and higher visual processing that are usually absent in artificial deep convolutional neural networks. And we're going to look at some of these properties in a bit of detail today. So where we finished last time, we saw that there are responses of face-selective areas of the human brain, in this case recorded with functional MRI, and here these areas respond to faces more than various other object types. So this is the area that guy was just having electrically stimulated. Okay. But faces are not the only type of objects that produce responses in specific brain areas. If we show subjects in MRI scanners pictures of objects, of faces, and of places of various types over here, we see very different responses to these. And this actually highlights a region fairly near the parahippocampal place area, again in the inferior temporal cortex. And this area, if we look at how it responds to all of these different things, right, it really doesn't have any response to faces at all. It has some response to objects, particularly when you group those objects to start making something that looks like a scene. Okay? And it has much stronger responses to things that look like buildings, to landscapes, and so on. So as things start to look more and more like a place, right, this area starts responding more and more. Okay. So we've now seen specific responses to faces and objects which are pretty similar to each other. Both of these activate specific areas on the bottom of the temporal lobe, the inferior temporal cortex, or IT. So here we see the fusiform face area here, and the parahippocampal place area, the area we were just looking at in green here. So Faces and places are particularly important for humans as we move around our environment a lot, and we're very social animals compared to other animals, so faces are very important for us. We also find an area called the visual word form area, which responds to written words. And you've seen in the labs that written words are often used, and digits are often used as an example of a type of object that we might expect our networks to produce a specific response to as well. So faces, places, and words are major categories in experiments on object processing. But to some extent, this approach is limited because the experimenters have used their own brains, so their own thoughts, to identify a type of object that they think is special, right? Like a face, a place, or a word. Okay. And then they tested whether the brain has different responses to that object and to other pictures that they can show. Okay. Now, they've used their brain to choose an object type, and then they found a response in the brain that corresponds to that object type, right? So perhaps it's not surprising then that if you can think of a type of object, then that the brain has a response to that type of object, right? Okay, but the experimenters have chosen what they think is the important thing about those objects. Of course, a picture of a face and a picture of an outdoor scene differ in lots of different complicated ways. All right, and we're really just looking at what the experimenter thinks is the important way that they differ. That becomes a bit of a difficult experimental design. Okay. In this experiment, which is one of the experiments on your reading list, the experimenters got natural movies, right, and then they labelled when in those natural movies different types of objects occur, and then they showed those movies to the brain. All right. So literally there, they just got any object, any noun that occurs anywhere in the movies, right? They tagged those. Okay. So that tries to get away from like picking one class of objects that you think is special, right? So then they correlated the time courses of the brain's response to the times that the objects were labelled in the movies, right? And then they determined the object preferences of their MRI recording sites in a kind of less biased way. So here you see a little image of the brain. Here you see it kind of flattened out or unfolded using a computer algorithm. And here you can see V1 is right at the back here. And what you see is that they see responses to all sorts of different objects. So they've put them into broad classes that have some relationship to each other. So for example, humans and body parts are these kind of blue-green colours, right? Animals are a bit like that, so they're a yellow colour. Right, but roads and vehicles are not so much like humans, so they're kind of further away. Right. I forget exactly how they kind of came to this scale, but that's what they ended up with. And so here we see the brain's responses to lots of different object types, therefore labelled with different colours. And you can see the parahippocampal place area here in red, right, and the fusiform face area here in green because it responds to human-like things. Whereas the parahippocampal place area responds to geography, outdoor scenes, that type of thing. Okay. So this shows that there are object selective areas for a large range of different object types. More recently, again this is on your reading list, the same group has revealed a network of areas that respond to specific different types of semantic content in tagged narrative speech and reading. Okay, so here they got podcasts, story podcasts, and they labelled, well, they got a transcript of the podcast. Okay, and they used that to analyse when the words came up in the sequence that they were playing, right? And then they could see what type of word, what type of semantic content in the words different brain areas respond to. And here you see the parahippocampal place area and the fusiform face area don't respond very strongly at all. And indeed, the areas that respond well in the last experiment, the visual object areas, don't respond very well at all. But a whole new group of areas, so at the junction of the temporal lobe and the parietal lobe, and also deep into the frontal lobe, start responding to semantic content. And in a follow-up of this, they've shown that this is regardless of whether that's shown as text or as speech. And they've done it both ways. So this is a particularly important type of experiment because we can't investigate linguistics very well in animal models of the brain. Okay, so we can investigate the structure of language and things like that, but it's very hard to look at the brain's response to language in an animal, because animals don't have language. All right, the closest that what people use instead sometimes is songbirds. All right, but songbirds are very, very different phylogenetically from humans. They're not close relations of humans like monkeys are. So we might expect very different responses to bird song to speech. So this is also interesting because we have little idea of how the brain processes language, and you'll see a lot about that in the second part of the class. But we do see that the brain reaches very similar results to processing visual objects. All right? So these responses that we see, they're in different brain areas, but we also see a separation of different types of semantic content. So this implies that deep learning mechanisms that we've already seen for object processing are likely to be involved in language comprehension as well. Okay. So this paper kind of ties together what we've been talking about with object processing with what you're going to see in the second part of the course. Right. So yeah, you'll read a bit about that later. So if we show images to some object selective area that here responds to baseballs more than it responds to faces, okay, we see that this area responds more when baseballs are shown and less when faces are shown, okay, as you'd expect. However, if we instead ask people to imagine baseballs and imagine faces without showing them anything at all, right, the same area responds when they imagine baseballs and responds less when they imagine faces, just like it does if they were looking at these things. All right. So object selective responses, therefore, don't just depend on what we're seeing, but also what we're visualizing or what we're paying attention to. All right. Indeed, if we imagine images, feedback on image content seems to go all the way back to the primary visual cortex. So in this experiment, the experimenters showed a large set of photographs, just random photographs taken from the internet, I believe, to subjects in an MRI scanner. And then they can analyze where contrast lay in those images and where the orientation content was in those images. And they can then make they can compare that to when each recording site in V1 responds, to derive a model of what each recording site responds to. So which positions and orientations in V1 the recording site responds to when shown actual photographs. If we, as one of the papers I've assigned to shows, if you then show unseen pictures, okay, so pictures that have not been included in the training set here, you can predict quite far above chance from the brain activity, what picture people were looking at. All right. By basically saying, well, what responses would the model predict to this picture that we're showing and to all of these other predicted pictures, okay, and then you compare those against what you actually see, and the one that fits best is the most likely to be the picture that you saw, that the person was seeing. But you can also train people to imagine these artworks. They used famous artworks because they're relatively easy for people to imagine. And here, if you show nothing, okay, you can again identify from the activity of the primary visual cortex what picture people were imagining. Okay, so this shows that imagination, or imagery, or visualization, produces quite similar responses in the early visual cortex to actually looking at something. So it kind of shows that the image representation from higher levels of our brain is imposed onto a precise retinotopically organized pattern of activity in V1. Right, so it's not just more or less activation in V1, it's a very precise representation of what's in the image that you're imagining. Okay. All right, so and back to this experiment briefly, where we see the responses to different types of objects. Okay. When mapping these object selective responses, the subjects were just passively watching the movies. Okay. You can give subjects a task instead, so they're not passively watching the movies. And here we're going to see what responses you see when you ask the subjects to press a button whenever they see a human. Okay? And here you see this nice rainbow of all of these different object preferences. But if you get people to press a button when they're seeing a human, so pay attention to humans, right, then you see all of these colours kind of shift towards the human end of this spectrum. Right? Similarly, if you get people to press a button whenever they see vehicles, so that's at this end of the spectrum, you go from these sort of yellow, green, blue colours to kind of more purple and red and yellow colours. Right. So here this shows that object selectivity in the brain depends quite strongly on the task that we're doing. Right? A face selective area can become a car selective area if given a car identification task. So what we're seeing here basically is that the responses of these neural populations are being attracted towards the thing that we're attending to. Okay. Similarly, we can attend to a specific spatial location, and this attracts spatial receptive fields towards that location. So here we see a bar moving across visual space to map out the spatial responses of all of the voxels in the early visual cortex. Okay. And we do this when subjects are paying attention here, they're looking here always in the middle, but they're paying attention to one of these two little patterns on the right. And if you watch these very, very carefully, this is a very difficult task. Every once in a while, these have slightly higher contrast than at other times. Okay. So this is a really hard task, and importantly, it's impossible to do on both sides at the same time. So you have to look here, but attend to one side. All right. So if we attend to, so if we're looking here, right, but we're attending to a target on the left or on the right here. Okay. If we have a spatial receptive field like this, right, and we're attending to the left, okay, this attended location on the left seems to draw the spatial preference of this receptive field towards it, like so. Okay. So this is the receptive field that you'd measure under the influence of attention to the left. Okay. Alternatively, if you attend on the right, this receptive field, which has the same sort of starting state, right, will move towards the location on the right. Okay, we can then compare these positions under these two attention conditions to see how attention affects the visual cortex's spatial representation, so how it affects the receptive field positions. So here we can see for the whole, this is the sort of breakdown of the whole visual field into different sections. All right, we've just got it for half of the visual field here. If we get one of these little sections here, and we get all of the recording sites in it that respond best in this section, and we say, well, where do they respond when we're attending on the left? It's that location. Where do they respond when we're attending on the right? That location. We can join them together, and that shows how attention on the two sides is shifting this spatial preference. Okay. So the spatial preferences don't seem to be fixed here. Okay. You can repeat this for all of these areas. So this is another little quadrant here. Okay, do the same thing. You can repeat this for all of these areas, and you see that over this whole visual field representation, when we attend on one side, all of the receptive fields are attracted that way. And when we attend on the other side, all of the receptive fields are attracted that way. Okay. So visual spatial receptive fields are not fixed, just like those object preferences. They're attracted towards the place that we're attending. Okay. You can see this in a group of different areas here. So this seems to happen all the way through the visual processing hierarchy. So all of these areas show shifts in the position of receptive fields under the influence of attention. In later visual processing, so here we see V1, the start of visual processing, and the IPS visual field maps, which are much later in visual processing, and are thought to be very much involved in guiding attention. We see that the change in these receptive field positions increases up this hierarchy. Okay. So we see a really small but measurable effect in V1 and a really large effect as we get to higher areas. It's important to notice when sort of interpreting this that the receptive field sizes also increase in a similar way as you go up the hierarchy. Okay. So it seems that basically in later visual processing, the neurons and the neural populations, as we're seeing here, are less sensitive to visual position, so they have larger receptive fields. Okay, and that means that their preferred visual positions can be changed more because they have a less precise representation of those visual positions. So what this does, basically, is if you're attending up here and you kind of see this as the visual field representation without attention, this is what happens when you add attention. Okay? So that you see a kind of zoom in on the area that you're attending to in the brain's representation of visual space. Okay, so to summarize what we've seen so far, the brain has responses to many different classes of object. Okay, so faces, places, words, and tools are the most commonly examined of these, but there are lots. There seem to be responses to all types of objects. Similar responses have recently been found for semantic content in language, okay, which suggests that similar processes are involved. Also, these responses can be driven by imagined content. Okay, so this gives similar responses to actually presented scene content. Right? And this goes all the way back to the early image representation in the primary visual cortex, because you can look at V1's activity and identify what image people were imagining. Similarly, spatial responses are drawn towards attended content. Okay, so object responses are drawn towards the attended object. Spatial responses are drawn towards the attended location. Okay, so this shows that basically this whole representation of both space and object identity is flexible depending on what we're doing. All right? None of that can happen in the networks that we've seen so far. Okay? These networks that we've seen so far take an input, they transform it, and then they give an output. All right? There's no way that imagined content or attended content could influence that process. So imagining an object will generally start by activating a higher level representation of that object, which then must be fed back to the early representation of that object. Okay. Similarly, attention is thought to be driven by higher level areas that are more involved in sort of task demands than early visual processing. So in these higher areas, goals and decisions are represented. Okay. And that effect of attention is then fed back to the early visual cortex. Right. So, the networks that we've seen so far have always this feed-forward structure. So they go from the image to the first layer to the next layer.
Do we see some sort of a signal that represents an engine telling everything to shift, or is it just that it does something?
Yeah. So there's basically it seems to be a control on gain. All right, of responses. So that is to say, if you show nothing, all right, and you get people to attend to one location, the neural responses are kind of zero because you're showing them nothing, right? And those are being multiplied by some gain factor, right? So that doesn't really have an effect when you're showing people nothing, right? But when you show people something, then the gain seems to increase the response to the thing that you're paying attention to. Okay, so this is known as a feature selective gain model of attention, which is to say that when you attend to something, the gain on the thing that you're attending to increases, and on the things nearby it also increases as well. Okay, so we've seen this network so far, this artificial network that kind of builds from early responses using always feed-forward connections going from one layer to the next to the next. Okay. But if we look at this biological network at the top, it has feed-forward connections, but also in dashed lines here, these dashed arrows, it has feedback connections as well. Okay. Each of these units is not just affected by the image, it's also affected by responses happening at later areas as well. We also see that each layer has inputs from units within the same layer, these kind of curved circular arrows that you see here. Okay. So these are called lateral connections, so that's basically connections within the same layer that modify the responses of one neuron based on the other neurons in the same layer. In artificial deep convolutional neural networks, generally neither of those things is implemented. Okay, we'll see a network where it is in a minute. And the final type of connection pathway, which is often found in artificial deep networks, is called a recurrent connection. Okay? So this is where, in artificial networks only, a neuron's activity depends on its own recent activity. That's what a recurrent connection is. Okay? That a neuron's a unit's activity depends on its past. Right? So it's time dependent. So in artificial deep convolutional neural networks, So in artificial deep convolutional neural networks, the input is always a static image, right, with no temporal information, or very normally that's what we show. Okay. So recent activity there will always be 0. Okay. But in networks with temporal inputs, recurrent connections are a very useful thing to use. Okay. So this is particularly important in language because language has a sort of temporal sequence structure. So it's useful to know what came earlier in the sentence to interpret what people are saying now in the sentence. Okay, so we've got various different connections that we've just talked about. We've talked about feed-forward connections, the normal type of connections, feedback connections, which are kind of sending signals related to later activity back to influence these earlier areas. We've also got lateral connections within the same layer. Okay, and in the context of artificial networks, we've got recurrent connections, which are a neuron activating itself, basically. So we've looked at various of these, and all of these are represented in this diagram in the case of inputs from the lateral geniculate nucleus of the thalamus into V1, which is this layer of neurons here, and then feeding on to higher cortical areas. So in blue here we see our feed-forward connection, so our inputs coming up from the thalamus. In brown at the top here we see our feedback connections, which are kind of inputs coming back from the next layer up in the sequence, so from this higher cortical area down here. Okay. We also see, so in artificial neural networks, recurrent connections, as I mentioned, typically activate the same neuron, right? But in biological neurons, neurons never synapse with themselves. Okay? So a neuron can't directly influence its own activity. But it can influence its own activity indirectly. So, for example, what we see here is we see a type of lateral connection where this neuron's activity will stimulate this neuron here, right, which will then feed back to inhibit this neuron here. Okay. Yeah?
Just a quick question from the slide before this one.
Yeah.
Basically, everyone says that backpropagation is a thing that artificial networks do, and that doesn't occur in biological systems. However, you do see like a stream going backwards. Isn't that something similar?
It is something similar, yeah. So, I was wondering whether to talk about this. It gets a little bit technical, so I'm going to keep it a bit short. But basically, backpropagation could occur in a biological system if you have symmetrical feed-forward and feedback connections. So, and that actually doesn't sound that surprising, right? So if you have symmetrical feedback connections, it means that you can alter something about the earlier neurons depending on what happens at the end of the sequence, right? But that needs to be in a very, very precise way, right? And indeed, it looks from some of these results like something like that might be happening. For example, if you imagine an image, right, you see a really precise activation of the early visual cortex, right? So it's possible that if you have sufficiently symmetrical feedback connections, that might produce backpropagation-like effects. It's occurring through a very different mechanism, so I don't like to call it backpropagation, right? But it might produce an effect that looks a little bit like that. Okay, so as I said, a neuron can't directly activate itself, so we don't. What we call a recurrent connection in a biological network is not really the same as recurrent connections as usually implemented in artificial deep networks. But this neuron that we see the interaction with doesn't have to be just this type of neuron. What could happen also is that we could have an excitatory connection from this neuron to this neuron and another one going back, so that they're both kind of pushing each other up, right? So that's another type of lateral connection. Okay. And it's also possible for neuron to, so let's say this neuron feeds some activity up to the next layer, right? And then neurons in the next layer feed back down to it. Okay, so you can have a neuron influencing its own activity through connections with any number of different types of neurons in the earlier layer, in the higher layer, or in the same layer. Okay, those can be excitatory or inhibitory interactions. Okay. as a result of this, right, all of these interactions that we see here, particularly these back and forth recurrent type interactions, they take some time to happen. Okay, so you know this. If this neuron's activated, it's going to activate this neuron and then send that signal back. Each of these steps has a little temporal component to it. All right. So therefore, in any type of activity where you have any type of network where you have activity going in multiple directions or in loops like this, right, then the network's response becomes inherently time dependent. Okay, and indeed, if you think about what this neuron's going to do, right, it's going to activate this neuron and then this is going to deactivate it, right, and then so this response goes down. Oh, sorry, I'll show this with the next slide. If we activate this neuron in the excitatory pool, all right, this becomes active. Then this is going to activate an inhibitory pool of neurons. Okay, so then this inhibitory pool of neurons becomes active, which then in turn inhibits this first pool of neurons. All right, so this deactivates, which deactivates that connection, deactivates the inhibitory pool. Okay, deactivates that connection, so this is no longer inhibited. So we kind of get back to the start that it's being excited by its input, right, and not inhibited by this inhibitory pool. And then we kind of see the same thing happen again. So all of this processing becomes very dynamic, right? There's lots of temporally unfolding interactions, and the neuron, instead of just getting an image and processing it, it tries to find a kind of equilibrium between different neural populations that are going up and down all of the time. Okay. So this cycle of excitation and inhibition that we see here is known as a neural oscillation. Okay. If we look at sort of various measures of electrical activity measured a little bit further away from the cell than spiking activity, like this, we see that the voltage in the brain tends to go up and down in these cycles like this. Okay. And indeed, during high parts of these cycles, there's lots of neural spiking. And during low parts of these cycles, there's less neural spiking. Okay. So that's a neural oscillation, and that occurs. So that's what EEG measures, for example. Right? And that occurs because of these back and forth interactions between excitatory and inhibitory populations, which can be within the same brain area, or it can be off to another area and then back again. And depending on how far away, the other thing is that might change the time constant and therefore change the frequency of this oscillation. Interestingly, actually, if you look in the low points of these cycles and you show people an image there that's difficult for them to perceive, the performance at perceiving that image during the low points of these cycles will be less than their performance at perceiving that image in the high points of these cycles. So these oscillations directly affect our perception moment to moment, but this just happens so quickly that we don't normally feel it happen. Yeah.
Flashing images in 0.1 seconds, you're like, what is this? Or how do you get?
Oh, you show it very, very briefly. So humans are actually, we have really great temporal perception. If you show a flash of an image just for one frame, right, which is, let's say, you know, even at 20 frames per second, right, that's only like 50 milliseconds. So we'll get kind of half of one of these cycles, right? We can perceive that perfectly well. But even if you drive a monitor at 1,000 frames per second, you just show one, the visual system picks up on it, even though it's shown only very briefly. All right. Here we are going to quickly look at recurrent activity as it's typically modeled in an artificial deep network. All right. So here this is primarily a way of dealing with time. All right. In a layer with recurrent activity, the activity of each unit at one point in time then feeds back to the next point in time. So effectively this means that the unit is partially activated by the past version of itself. Okay? And this link between a neuron and the past, a unit and the past version of itself has a weight, and that weight can be learned. All right? Just like all of the other weights in this network. All right? So this is useful, as I say, in lots of tasks where temporal sequence is important. And so while spatial relationships are very important for visual processing, temporal relationships are very important for sound processing, language processing, things like that. So if we unfold this over time, basically we've got unit at time point t-1, which then sort of feeds, and this is the same unit, right, but is also potentially changing its activity over time because the input is changing over time, right? So different parts of the sentence, for example. Okay. Here we can see that the past activity then it feeds up to the next layer, but it also feeds into the future, and then feeds into the future again. Okay. So some examples where this is useful is in text-to-speech synthesis. So how we say a word at this moment depends on what we've recently been saying. So recurrent networks are very useful for synthesizing convincing sounding speech. Okay. Also for speech recognition, so we were giving a temporal stream of information and we want to determine what that's about. So that's a very useful thing to be able to do. And also in semantic comprehension in language, this type of network is very useful. And finally in language translation. So here this also works with text, of course. It's just that the sequence in the text is kind of replacing the time aspect here. But in language translation, I don't know if you guys are old enough to remember when online translation was awful. Online translation used to be really goddamn awful, and I noticed this a lot when I first moved to the Netherlands and didn't speak Dutch. But now, if you throw a document through Google Translate, the translation is pretty accurate, and that's because they started using deep recurrent networks for doing it. Okay, so as I say, this is a real simplification because a neuron is always kind of activating itself, or at least neurons at least in the same layer are activating themselves, okay, or activating other neurons in the same layer. Here is the first artificial network to incorporate these extra connections that we've seen. So here we've got a bottom-up connection. So this only happened in 2017, the first one of these. And this is, again, as a tool for investigating visual processing. Okay. So this is again driven by cognitive neuroscience. So it's trying to imitate what's going on in the brain. Okay. So the networks that we've looked at in the past are of this B type, so bottom-up, right? The input goes to one layer, then the next layer, then the next layer, and that makes a decision. Okay. We can also implement feedback connections. We can also implement recurrent connections only, or both recurrent and lateral and feedback connections. All of these, this is why I just said recurrent there. All of these connections in biological systems where you've got feed forward and feedback, or lateral connections which will then affect the state within the layer and then affect the neuron's activity back again. In biological systems, those are all globally referred to as recurrent connections, okay, which is an important distinction from artificial deep convolutional neural networks, where recurrent connections really mean one specific thing. So this complex type of network, where you have bottom-up, lateral, and top-down connections, or BLT, is a very interesting recent network that's just been developed. So it's pretty straightforward to make a convolutional filter that doesn't only sample from the previous layer, but has parts of the filter that go up into the next layer, right, and parts of the filter that are in the same layer. Okay. So it's pretty straightforward to program something like that. Okay. But that convolutional filter needs to be larger because it's got aspects, it's got elements in these different layers, right? And so more weights need to be learned in a network like this, because you have the convolutional filter going down, convolutional filter going up, and the one in the same layer, and these are all one and the same filter, basically. Right. Now, in a network like this, again, the activity in one unit can affect another unit, which affects it back. Therefore, we don't only have one point in time, right? There's lots of time steps involved in this, right? And the neural activity is going to go back and forth to try to reach some type of equilibrium. So, perhaps more complex than the state of the convolutional filter going into higher layers and lower layers in the same layer, right, is that the network's activity will move back and forth as a result of each interaction effect and the activity the changes that has on the interacting element. So here we can see this basically separated over time. So at the first time step, there's no activity in these higher layers, right? So we've just got the activity of feed-forward connections, right? But then at time point 2, the activity in the first time point at these higher layers and in the same layer is going to also affect the activity in each of these layers. Okay? So this becomes an inherently time-dependent network. All right? And this is perhaps the more complicated thing about it. All right? And those two things come hand in hand. Right? If you have any type of activity that can influence some other type of activity that might influence it back, right? Inherently the network becomes time-dependent. So here the experimenters have simplified this. They use a fixed number of cycles. Here they use 4 cycles. Okay. Of course, the brain doesn't have discrete time steps, and there's no end to the interaction cycles, unless you make the person unconscious. All right. The input image, in this case, remains the same through all of these cycles, so the same input is maintained. Though, of course, in biological systems, we live in a moving world. We're always moving our eyes, and the world's always moving around. So this keeping the input the same is, again, an oversimplification. All right. So if we look at what these networks do, they've tested them on a digit recognition task. All right? And as you've seen in the labs already, probably, this is a pretty easy task. A relatively simple network can achieve a digit recognition task. It doesn't require very deep networks. It doesn't require very complex networks. But this task becomes very much harder if you add parts of other digits in here. So here we've got a kind of scramble of various different digits. So this is kind of debris, as they call it in the paper. And then they add that on top of the digit that they're trying to identify. Right? This looks a lot like a CAPTCHA, the sort of thing to identify yourself as a human online. Right? And so basically a human can identify this pretty well, but artificial networks generally do very badly at this type of task. So basically what they've done is they've made the digit identification task really hard, but humans can do this perfectly well. Right? So when they do that, if they have very little debris here, okay, so that's in dark here, then the error on the classification, even of a bottom-up network here, is pretty low. Right? But as they add more debris here, then the error on the bottom-up network gets very high. Right? So this becomes harder and harder for the network to identify the thing. Though humans, again, can do this pretty well. Okay. If we look at the BLT network, the bottom-up, lateral, and top-down network, then, first of all, even under low debris conditions, so these dark bars here, right, it performs much better. Right? But particularly under high debris conditions where there's more noise added to the system, it really performs quite well, whereas just a bottom-up network does very poorly. Here this is also separated into bottom-up and top-down only, bottom-up and lateral only. Right? And these two, the BF and the BK conditions, are both bottom-up only networks, but here the filters are larger, or there are more filters, so that's those two different cases, larger filters, more filters, so that the same number of weights need to be learned in the network as in a more complex network like this. Right? So even though, so these two are particularly well controlled, that basically the same number of weights need to be learned. We have way more filters per layer in this case, and here we have more complex filters spanning separate layers in this case. And here, even though it's very comparable to this, except for its lateral and top-down structure, we see that this network doesn't perform so well, and this one does perform much better. Okay. So these clearly there are influences of the feedback and the lateral connections, and these clearly improve performance here. Similarly, when we need to recognize multiple digits, so that's what we see here, we've got sets where there's several targets in the same image with the digits overlapping with each other here, and it can be more and more digits. Again, this is something humans do perfectly well. We can identify what these are very easily. Right? But bottom-up network actually is generally pretty bad at this, even with only 3 digits, and really pretty bad with 5 digits. This is a very good, so on here we can see the error here is 70%, on this scale it's 20%. Okay, so this is a much, much smaller error, so bottom-up networks can do this relatively well. Right? But when we include lateral and top-down connections, again this is doing, this network is doing much better. It's not doing so much better than this sort of well-controlled comparison here. So this is a network with more filters. So there is definitely an improvement between these two, but it's not huge. When recognizing handwritten digits, so here we see handwritten digits from the MNIST data set, which is again something that's included in your labs, we can see again that the BLT network is doing pretty well, even after we add a lot of noise to the target here. Okay. While a bottom-up only network does less well at that. So in this case, if you look at the bottom-up and top-down network here, the BT network, and the BLT network, these are doing very, very, very similarly. Okay. So here it seems that the top-down connections become very useful for disregarding noise in the input. Okay. And this is basically probably because the noise that they've added is relatively fine-scale noise, right? Whereas the target is a large-scale target. So if you look at the target, this image here, right? That's a really large-scale target, so later areas that are integrating more spatial information are likely to have better information about that target and then feed that back. Okay, so to summarize this part, the brain relies very heavily on lateral and feedback connections. Recurrent activity is implemented through both lateral and feedback connections. So a cell's activity can affect itself through feed-forward feedback interactions and through lateral interactions in the same layer. So this results in a dynamic network which has neural oscillations, basically. So there's no fixed state of activity here. In artificial deep convolutional networks, recurrent activity is usually implemented by a neuron activating itself, okay, which is a simple way of representing time and memory for time-dependent tasks. So, for example, language processing, where this will come up again. Biological neurons, on the other hand, never synapse with themselves. Recent experiments using artificial networks to investigate lateral and top-down connections, this works perfectly well. It's just more computationally intensive. Right? So the resulting interactions do make the network inherently time-dependent, so that becomes more computationally intensive because you have to model multiple time points. Right? But the image classification performance, particularly in difficult tasks, improves considerably. Now, this is more computationally intensive in two important ways. First of all, the filter becomes more extensive because it needs to go into the lower layer, into the same layer, and also into the higher layer. And also, we need to model multiple time steps in order to achieve this, right, which is a different type of problem. Okay. Question at the back? Sorry, let me just close this door. Let's try that again.
Given this
So, let me get over here so we can record what I'm about to say. Yeah, so indeed, it does become, given this sort of feed-forward, feedback nature of biological visual processing, it does become useful to start thinking about visual processing instead of in the sort of feed-forward way of analysis of an image. To think about it in terms of perturbation of kind of what the brain has seen before, right? It is often thought that the brain really is representing differences from its expectations. Right? So that's known as a predictive coding account. Right? And I think is a very useful way to think about it, is basically that what the brain does is it has expectations of what's going to happen, and indeed, everything that you see, right? You don't generally take a face or an image that you've seen before and break it down and analyze it, right? You know what that object is, right? So you know what it looks like, and you don't need to do this whole complicated analysis. As soon as you've recognized it, you can stop thinking, right? Whereas if you see a new object that you've never seen before, you're going to look at it for a while, it's going to take you a while to think about, right? So a very useful way of seeing visual processing is that it kind of analyzes the difference between what you expect to see and what you're actually seeing. Right? And visual processing is very low in predictable environments and very high in unpredictable environments. So in that way of thinking, indeed, the brain is really representing the error on the representation. Okay, so it becomes a bit like backpropagation of error. Okay. All right, so at this point we're going to take a 15-minute break. And so we started the last lecture by introducing the visual field map layout of the visual cortex, right? So we saw in the visual cortex that the brain has a representation of visual space where spatial position preferences gradually vary across the cortical surface. And that has certain computational benefits, as we saw, right? So in this image, we see different colors representing different positions in the visual field, here in two different coordinates, in two different coordinates, in a eccentricity direction and in a polar angle direction. And so here we can see the sort of map organization of the primary visual cortex, which all of this stuff built on. And so within these maps, right, we can see the responses of each fMRI recording site here gradually changing across the brain surface. So we can think of this as being a map of visual space, and that's generally how it is thought of. All right? But in fact, these maps don't really represent visual space directly. Instead, they represent a position on the sensory organ, that is to say the retina. All right? They represent a position on the retina, not in the outside world, because the input is the retina. Okay, so the input is, again, not an image, it's a pattern of neural activity in the retina. Okay. So for the human brain, then, visual field position is perhaps better understood as a location on the retina. So outside of sensory systems like these, we still don't have a good understanding of how the brain processes and organizes information. So generally, in more cognitive processing, we have ideas like, yeah, this blob of area, this like little continuous area of brain, lights up when you show people faces. All right? We don't really know what's going on inside that. We don't know its internal organization very well at all. In fact, mostly it's a visual field map, but that's another question. So what I do in my research is to look at how the brain's cognitive functions are organized. So how the brain processes and organizes more high-level cognitive information. Right? So when we started thinking beyond these primary sensory attributes, so here I mean things like visual position, position on the body, auditory frequency is the other really important one here. So for audition, the brain has a map of auditory frequency, and that's partly because the sensory organ for audition, the cochlea, is organized by auditory frequency. So there, in all of those cases, right, we see that the brain is just following the structure of the sensory organ. Right. When we started thinking beyond these sensory cortices, it seemed quite possible that such map structure could only follow sensory organ structure. Okay? Because the neural pathways that are coming in from the sensory organs are already organized as maps. Alternatively, it could be that responses to cognitively interesting features emerge, and then those might self-organize into a map organization, right, to allow patterns within those features to be analyzed effectively following the same principles that we've seen before. So it could be that we only have this type of map organization where the incoming information is already organized like that. Right? Or alternatively, it could be that the brain makes this map organization for efficiency, and therefore this could emerge for features that are extracted by the brain. Okay. So cognitive functions, right, are appealing because they're often absent or very different in animals. So to understand the processing underlying cognition, it's really best to measure it in humans. So I started looking in this direction by looking at responses to the number of visual objects that we're seeing, or numerosity. Okay. And this is a useful thing for humans to be able to perceive. Right? So if we see a display like this, okay, we can see a little group of objects, in this case dots, very simple objects. And we know how many objects it is pretty accurately, particularly with small numbers of objects. All right? And so up to about 6 objects, humans can enumerate a set like this very quickly, very accurately, and very confidently. And this is, so humans think we're clever with numbers, but a newborn chicken can do this, right? So this is not necessarily a very high level of cognition. And this allows us to make decisions between greater and lesser options. So if you are a foraging animal or a person shopping in the supermarket and you have these two choices, right, knowing the number of objects is obviously a useful thing for us. And we can make a decision based on this. So this is a really useful thing for us to be able to do. This is also interesting because for quantities like this, this is also affected by other quantities. All right? So if your decision becomes that, right, if you're doing kumquats versus grapefruits, right, you might make a different decision. Okay. So we started by looking at quantities for this because quantities have a straightforward structure to them. All right? But we don't have a sensory organ for quantities. Right? We have to derive responses to quantities. Okay? But there's a very clear structure. Is there more stuff or is there less stuff? So we started looking for maps of quantities. And we think that this might be a really good starting point for certain types of cognition, particularly quantitative cognition, that is to say things like mathematics, engineering, and more generally having like an internal physical model of the world. So we started looking for maps of these following the sort of relatively obvious prediction that map structure would follow the level of quantity. Right? More stuff might be represented at one end of a cortical area and less stuff at another end of the cortical area. So this would, of course, group similarly responding neurons, just like in the early visual cortex. And importantly, it would allow analysis of those mapped populations of neurons using deep convolutional neural network type methods. And for us as experimenters, this is convenient because in a map like that, neurons with similar responses are grouped together. So if you want to measure those responses using functional MRI, okay, which has a limited spatial resolution, if all of the neurons in a small area are responding similarly, then you can characterize their responses, and that will tell you a lot about the response of single neurons within that area. So that's like a practical thing that this buys you if you have this map-like organization. Okay. So we already knew some interesting things about number perception from animals. So, well, also from humans. In behavioral tasks here, so this is just you see a pattern of dots, and then you see another pattern of dots, and you're asked, are these the same number of items? When we see 2 dots, and then we're asked if 1 or 3 is the same as that, we basically will never say that 1 is the same as 2, and we might occasionally say that 3 is the same as 2. But if we show 6 dots here, okay, and then we ask the subject, well, is 5 the same as that, or is 7 the same as that? Quite often they will make a mistake there and say that 5 and 7 match 6. Okay. This is actually done in monkeys rather than humans, but you see a very, very similar thing in humans. So basically, as we increase the number of items, the accuracy declines. Right. We also know from monkeys that there are neurons that give tuned responses to numerosity, where there's a particular preferred number of items, so in this case 1. And with distance away from that, the response decreases. Similarly, we have here a neuron that responds most to 5, and with distance away from 5, the response decreases. And we also have neurons that respond to 3, and then the response will decrease on both sides. So using some fairly fancy functional MRI methods that I'm not going to go into, we basically mapped out the responses of MRI voxels, MRI recording sites throughout the brain, to changing numbers of objects. So we showed numbers of objects that change gradually, and then we mapped out where we see the peak response, and that tells us for each recording site which number of objects it responds most strongly to. So if you do that, and then you color code on the brain here, so here we've got the brain surface, and here we have a color code where different recording sites are colored by the number of items that they respond the most to. If you do that, you see several areas where, as you move across the cortical surface, we move from low number preferences to high number preferences. So we see a map-like organization. You can quantify this a bit statistically by saying, well, if we go from one end of this map to the other, how does the average preferred numerosity progress across the map? So this shows us that maps of cognitively interesting features, like the number of items we're looking at, emerge in the brain to represent continuous feature spaces like quantities, even though those don't follow the structure of the sensory organ. So there's no way that this structure is just inherited from the incoming neuron structure into the brain. Okay, this has to be computed in the brain, and then the brain self-organizes these responses. So that shows that even in cognition, right, we start having a similar pattern of organization which might have similar computational benefits for the brain. We've since extended this approach to start looking at event timing. Humans have a very interesting group of skills with timing. So humans can combine our timing from our senses of vision, hearing, and touch into a unified perception of time. Okay? And then we can make skilled movements that follow this timing. To put it simply, animals can't dance. Right? Yeah. Dancing relies on seeing what your partner's doing, hearing the music, feeling what your partner's doing, and then making skilled movements that follow all of that timing. So despite the importance of timing in perception, in multisensory integration, and in action planning, time has proved to be a very elusive thing to study in the brain, and it's still a really open question how the brain processes time. So the mechanisms of time processing in the brain are only beginning to be understood. So there are some similarities, though, between time perception and space perception and number perception. Okay? And so we kind of came to the hypothesis that the processing of event timing might rely on similar mechanisms to what we've already seen for visual space and for number. So what we did here was we wanted to test the idea of timing-tuned neural populations using functional MRI. So we got a stimulus where we could gradually vary two different parameters of the timing. So in this stimulus, we have an event happening once every second. So a dot appears and then disappears once every second. But the time between its appearance and disappearance is gradually varying. So increasingly, that 1 second that it's on between flashes becomes more and more full. Okay. Alternatively, so there we've got a constant period, so a constant frequency, and we're varying the duration of the event. In this case, we have a constant duration, so the event is always a 50 millisecond flash, but we can gradually vary how often that flash happens, so we can vary its period or its frequency. And in the third case, when one dot disappeared, the next one always appeared immediately on the next frame, which means that there's always a dot on the screen. It means that luminance, therefore, is constant, because there's always one dot on the screen, so that if you take just one time point, that will always look the same. Whereas duration and period are both varying together, okay? Because when one event ends, the next one begins immediately, so those two are very tightly linked. So we then analysed the responses to these changes in timing to see how neural populations in the brain respond to the timing of events. This is a very extreme case of some very, very fast timing. So if we project the preferred durations and periods onto the surface of the brain, we also see that various areas in the brain contain a range of duration and period preferences. So we have everything going from kind of yellowy colours up to purple colours. So from short, well, this is period and this is duration. From short duration preferences to long duration preferences. Similarly, from short period preferences to long period preferences. And again, we can quantify this by measuring the change in preferred duration or period with distance across the brain. And again, we see these nice gradual progressions. So within these areas, the preferred duration and period gradually and repeatedly change across the cortical surface, forming a series of map-like structures of neurons with gradually varying preferences for event timing. So, to summarize this, right, representations of visual position, body position, and auditory frequency have long been known to be spatially mapped onto the cortical surface, and these follow the structure of sensory organs. This allows analysis of local features within those maps using spatially limited convolutional filters, as we've seen a few times now. Representations of object number and event timing are also spatially mapped onto the brain surface, although there's no sensory organ with a structure like that. So this shows that basically that can emerge within the brain. And this would allow analysis of quantities by similar neural operations to what we've already seen in deep convolutional neural networks analyzing visual images. This basically means then that the representation of quantities can become the starting point for deep convolutional neural networks that analyze quantitative information, right, which might well underlie a lot of our abilities to do kind of higher cognitive functions that rely on quantity perception. So things like mathematics and having an internal model of physics. Okay, if you're interested in research projects in this direction for your research thesis, feel free to get in touch when that comes about. So far, we've just been looking at this for visual event timing. We're currently looking at this for auditory event timing. And of course, auditory timing is very important for things like music. So that's the type of thing that we're going to be looking at in the coming years. As this is the last lecture I'm going to give you, I'm going to finally direct you to the reading list that we've given you. So at the end of the last class, I brought up that paper on the representation of visual space in the early visual cortex and on reading spatial information and identifying images by looking at visual cortex activity. This paper does a kind of similar thing. It's actually from the same lab as that first paper, but here it does it for object types. So this is the experiment that we saw here, where different locations in the brain are responding to different types of objects. This paper, which is also on your reading list, there's a review article that goes with this if you find this paper very challenging, which I know a lot of students do. The review article is optional. I want you to understand this paper. This paper is the paper behind this. Okay? So behind this artificial deep convolutional neural network that is trained to do object identification and following the structure of the human brain, okay, which does very well at object identification, as we saw before. This paper analyzes the responses of the brain during dreams and identifies what people are dreaming about by looking at their brain activity, which I just think is super interesting. It's a little bit off topic, but it kind of shows this idea that feedback, or even in the absence of a visual stimulus, you see activity in dreams that looks a lot like activity during visual perception. And this paper looks at, again from the same lab, from the Gallant lab, looks at the structure of semantic maps. So this is what we saw for language processing. Okay, so here they look at the representation of language in the brain. Okay, so that's just like the main papers, a quick review of those and what those are about and how those relate to things that we've already seen. So that's it from me. I'm going to hang around for another few minutes if anyone wants to ask any questions. But from now until a few weeks, I don't remember exactly when the sort of part 2 lectures are going to start on linguistics. Please get through the labs and finish those and read these papers, because they're also going to be covered in the exam at the end. Okay. Does anyone have any questions before we wrap up? Okay, great. Well, good luck with the labs.